Hi everyone, welcome to a new video. Today we're going to be learning about proteins. Mind my voice, I am feeling quite under the weather right now, but we are going to get through this and we are going to know a bit more about proteins today. This is based on the AQA spec by the way, and it's based on spec point 3.1.4.1. So let's go over some key definitions. First of all, primary structure, which is the sequence/order of amino acids. Secondary structure, which is the folding of a polypeptide chain due to hydrogen bonding between amino acids. Tertiary structure, 3D folding of a polypeptide chain due to interactions, hydrogen ionic and disulfide bonds between R groups. Quaternary structure, multiple 2+ polypeptide chains. Dipeptide, formed by the condensation of two amino acids. Polypeptides, formed by the condensation of many amino acids. An amino acid is a monomer from which proteins are made, and a functional protein may contain one or more polypeptides. So firstly, let's look into the monomer-polymer side of proteins. So in a protein, the monomer, as you may know, is an amino acid, whereas the polymer usually has the suffix of peptide. So it can either be a dipeptide or polypeptide. Di obviously suggesting there's two amino acids, as mentioned before, and poly is just multiple. And a polypeptide is usually can be used in conjunction as a protein in most of the the mark schemes. So let's go over the general structure of amino acid. As you can see in the video, this is coming straight from the exam board spec, but if you can't, the general structural formula is RCH and H2 COOH, and to break down each of the substituent groups besides the hydrogen, of course, the R group represents a side chain, which is what difference between the 20 different amino acids, and that is a fact that you will need to know, that there are 20 different amino acids common within all organisms. The COOH group is also called the carboxylic acid group or the carboxyl group. It depends if you do A-level chemistry. As I mentioned a lot, you can use either term, and I'm sure it's a similar effect in A-level biology. This is where the acid part of amino acid is derived because it is an acidic group. The NH2, this is an amine/amino group. Once again, they essentially mean the same thing. On the specification, it is listed as an amine group though. And this is where the amino part of the amino acid is derived, and this is a basic group. The characteristic bond within proteins is a peptide bond, and the test for peptide bonds is called the Biuret's test. Once again, I'm sure people pronounce it differently, but I'm sure it gets the point across either way. And how to undergo this, you add Biuret reagent and mix, and if a protein is present, you will see a color change from blue to purple. And now we are going to be drawing how to draw a peptide bond. And if you're on audio, we are going to skip past this part because obviously you can't see the visuals, but if you do want to see the visuals, hop onto YouTube and you will be able to see it. Without further ado, let's draw a peptide bond. So we're going to draw, let me get a black pen. That's a pencil. I don't usually like pencils. Okay, so we are going to draw a peptide bond. So with this, we're going to draw two amino acid monomers. Okay, and as this is a condensation reaction, I always circle around the water that is removed. As you see, just for ease, I split up the NH2 as it would be drawn on a normal, like, what's the term? Display formula. So showing all the individual bonds. And you can similarly do the same thing for carboxylic acid group. It would appear like this. Just imagine that's like from this carbon from the main carbon chain. So now drawing the carboxylic acid group, it would appear like this. And then you could just circle around those two. But we're gonna add those. Let me see on the right page. And then here, I'm gonna add my H2O group first, and then I'm going to draw the final dipeptide structure. HCO. I like to draw my peptide, pardon me, my peptide bonds with like the CO group going up and then the NH group going down, but honestly, it doesn't really matter. And let me not forget the double bond right there. And this right here, let me highlight this. This would be the peptide bond right here. And similar to scientific drawings, I'm just gonna get a straight line, straight in quotation marks, of course, and label this peptide bond. And this structure as a whole, right here, let me get my laser pointer. This would be a dipeptide. Anyways, now let's get into the main body of the content. So when we're talking about protein structure, we're looking at a polymer of amino acids joined by peptide bonds formed by condensation. Primary structure is the structure/order of amino acids. Secondary structure is the folding of a polypeptide chain due to hydrogen bonding between amino acids. Tertiary structure is the 3D folding of a polypeptide chain due to the interactions hydrogen ionic and disulfide bonds between R groups. Quaternary structure is multiple 2+ peptide chains. So when we have a further look into protein structure, let me get my laser pointer. So here with the primary structure, I like to consider it like necklace with beads, and each bead would be an amino acid, as you can see on the diagram if you are looking at the video, and you can see what is circled individually in one of these blue circles is just an individual monomer of the chain, pardon me. And then for secondary structure, this occurs between the NH of one amino acid and the COO of another. And secondary structure, you can have alpha helices, as you can see. Let me zoom in on the diagram. You can have alpha helices that are kind of that helical shape we're so used to, and then you can have beta pleated sheets, which I like to think, I don't know if you ever watch the movies, but I feel like they always have like dressing rooms, and when they have dressing rooms they always have like these pop-up blinds where you can't see people, and I always just imagine it to look like that. So that's just like a funny way to remember it. Moving on to tertiary structure, for example, some proteins that contain this structure, you can consider the active site of an enzyme, receptors, intrinsic proteins, which include protein carriers and protein channels, which we will be looking into further throughout the topic. And as you can see, I really love this diagram because you can kind of see how the tertiary structure here, it's just one polypeptide, as you can see, but you can also see each like beta pleated sheet and the alpha helices within it from the secondary structure. So that's really, it's a good visual note. For quaternary structure, these may be associated with non-protein prosthetic groups. We'll get into that more when you look into hemoglobin. Each polypeptide chain is referred to as a subunit, and those with prosthetic groups attached are considered conjugated proteins. And some examples of quaternary structure proteins are antibodies and hemoglobin, as I mentioned before. And yeah, let's move on. So other bonds, So let's look into the other bonds that you can find within protein structure. So let's first look at hydrogen bonds. These are easily broken by pH and high temp. They're numerous. In secondary structure, mind you, they are between the NH of an amine group and COO of a carboxylic acid group. In tertiary structure, they were between the R group and an R group or amine or carboxylic acid group. Ionic bonds, they're easily broken by pH. In tertiary structure, they're between the amine and the carboxylic acid groups. Disulfide bridges, these are not easily broken. In tertiary structure, these are between a sulfur atom within the amino acid cysteine R group. And in terms of strength, it would go from disulfide bonds or bridges to ionic bonds to hydrogen bonds. So with that, we are finished this quick revision video on proteins. I hope this was a really good recap, but equally this can be a great starting point for your revision. So always come back to this video if you ever need any help, and obviously comment down below if you need any extra advice on the topic. But yeah, happy revision.